Close that. Yeah, so good afternoon, everyone. So, like I said, there will be an opportunity to have questions and answers at the end of the presentation. But again, thank you for joining at the lunch break. We're super excited to tell you what we do here at C3i. And we have one of sort of the North American specialists who will be presenting to you today that we're lucky enough to call a colleague and a friend, which is Richard Valencourt. So I'll just give you a quick overview about what we do here at C3i. So C3i was a company that was spun out of the Hôpital Maisonneuve at Rosemont here in Montreal, which is about 20 minutes from an international airport on a good day. So we're a full service CDMO that does everything from development to commercialization. So Richard, if you don't mind skipping on to the next. So I could read this, but really our mission statement is we're there for the patients. We have people on staff who were there for the very first CAR T cell therapy, actually. And I think we all have the same mission. There's people who've worked at the company for 15, 20 years and really see face to face the patients and know sort of uh, the difficulties with cancer treatments and the impact that has on themselves and the families. So, next slide, please, Risha. So, I think for business purposes, just briefly, we can split the business into three main sections. The first one is the GMP manufacturing and technology transfer. The next is the analytical testing, and the third part is supply chain logistics. So, if you don't mind just going on to the next slide, Richard, thank you. So we were lucky to have a couple of big milestones in the last 2 years. The first was the drug establishment license, which is the commercial license for manufacturing cell therapies here in Canada. We were the first one to receive it. And then more recently, we got our commercial manufacturing license from the EMA, excuse me, the European Medicines Agency. I'm sure Richard can tell you more about that than me. So what that means is we can do everything from development to commercialization for cell therapy and gene therapy manufacturing. The facility itself is a 40,000 square foot facility with 14 clean rooms. I think it's 13 grade B clean rooms and one grade A. And we produce about 500 units per year. So that's sort of a high level overview of what we do. Next slide, Richard. So if you do have any comments, questions, or need any of those services that I've briefly gone over there, you can email me, which was in the first part of the first slide that I presented, or you can use the business dev at C3, center3i.com. But without further ado, I want to introduce my colleague, Richard Valencourt. I'll let him do his own introduction. But Richard, we've worked together now for probably about 6 years, and he really is one of the best in the industry. So I'm super excited that he agreed to do this presentation and try and transfer some of the knowledge and mistakes and successes that he's had throughout his career, and especially C3i. So Richard, I'll pass the mic over to you. And if you can just give a brief introduction about your background and what you do here before you get into your presentation, I'd appreciate that. Again, just for everyone listening, there will be an opportunity for question and answers at the end. You'll see a pop-up with question and answers. You can submit them in French or English, and we'll get Richard to answer those for you. So, Richard, the floor is yours.
Perfect. Thank you for the introduction, Jamie. So I'm the Quality Assurance Director here at C3i. I've been with C3i in the quality department for the last 3 years. And before that, I was with the Maisonneuve Rosemont Hospital in the cell therapy lab doing starting validation and then evolved through various positions in the quality department. So when I was first asked to give that presentation today, I was wondering who would I address this webinar to, and then I thought about young me at the beginning of my career. But as I just said, I started in a cell therapy lab where we're doing mostly hematopoietic stem cell transplants. And then we had clinical research programs and manufacturing programs to enter clinical trials. So we thought we had to start about looking at the GMPs and see how we can comply with them. So really my first impression was a lot of confusion. It felt like all these guidelines were hard to apply to what we were doing, and that I had a very hard time picturing how we could be fully compliant to the GMPs. And then a way I saw it was like trying to fit that square block into that triangle shape. It was just impossible to me at the time. So today what I'm going to do with you is that I'm going to show you how we can fit that block into that triangular shape. So I'm going to give a very brief overview of the different guidelines that are applicable for manufacturing for cell and gene therapy. And then I'll focus on the main challenges that are associated with the manufacturing of cell and gene therapy products. And then I'll give you some maybe advice to how you should be preparing for an inspection. So back to my analogy of that square that we need to fit into that triangle. So I think the best way to picture is that all the GMP guidelines that are out there and that are applicable to cell therapy can act like a series of filter that will carve that block into the shape we want. So the first layer, so to speak, is the general GMP guidelines that are applicable to all drug manufacturers. Then the second layer is sterile manufacturing guidelines because cell and gene therapy products needs to be produced using aseptic techniques due to the nature of the product and the starting materials. Also, it shares a lot of similarities with biologics, biological drugs. So the guidelines that are applicable to these drugs are very useful for cell and gene therapy manufacturers. And finally, yeah, you have guidelines that are specific for cell and gene therapy. So in the end, once you apply all these filters, so to speak, or once you apply all these different layers of guidelines, then you finally can fit your big block into your triangular shape. So the first layer is the general GMP guidelines. Really what these are, it sets the base for what your quality, pharmaceutical quality system should be like, and it reminds you of the objectives. So the objective of your PQS is to deliver a product that has the quality attributes that will meet, so that you have a product that will meet the patient, the healthcare practitioners' needs, so that it's fit for purpose. You also, it also forces you to establish and maintain a state of control over all of your operations. And it's there also to facilitate continuous improvement because you want to remain in that state of control. And the basic elements of the PQS is your staff. You need to have trained staff to do your operations. You need to have premises that are suitable for your manufacturing premises, but also your utilities, so everything you need and equipment, everything you need to manufacture your products. You need to show control over your materials. That's very important. You need to have written procedures and control of records to make sure that you do what's written and you write what you do. You need to have consistent and robust processes throughout your facility. These need to be validated to show that you have the right controls over all your systems and processes. And you need to test your final products. So I won't go into too much details, but these are the general GMP guidelines for the FDA. On the US side, you have the Code of Federal Regulations parts 210 and 211. In Europe, you have part 1 of the GMP guidelines. Health Canada has guide 1 for good manufacturing practice. And the ICH has chapter Q10, which describes the quality system. And then the second layer is really sterile manufacturing because your starting material cannot be sterilized because your final product is injected into patients. So you need to maintain sterility throughout the process. So these guidelines focus on clean room design, how to use barrier technologies, isolator, biosafety cabinet. There's guidelines regarding gowning of operators in these aseptic suites. Obviously, how you manage aseptic technique, how you validate that process, and also very important, how you control and validate your sterilization if you can sterilize. So the guidelines for sterile manufacturing include the guidance for industry from the FDA for the manufacturing of drug by aseptic process. EMA Annex 1 is very useful, and Guide 119 from Health Canada as well. And then the biologics guidelines for biotical drugs focus on the challenges that are associated with biologics, mostly the higher variability that is often seen with the making of biological or biotechnological drugs, because the processes, the starting material are more variable. There's also an increased chance of having adventitious agents like viruses that can contaminate your products that will come from your materials, and also microbial contamination is a concern and is addressed there. So the biologics guidelines for the FDA, you have Part 600. For Health Canada, you have the Annex 2 to the GMP. And the ICH has a series of chapters, Q5A to Q5E, which contain a lot of useful information depending on what is your process. For cell lines, for example, a few of these chapters are very useful on how to manage them. And then finally, there are some guidelines that are specific for cell and gene therapy. Probably the most comprehensive is the European Part 4 of the GMP. So it's a guidelines for advanced therapy medicinal products under which cell and gene therapy products fall under. And the FDA has a series of guidelines. I think one of the most interesting is the CMC for gene and cell therapy. And other useful guidelines to consider. So everything that regulates the use of human cells, tissues, and organs. Although cell therapy products are not under the scope of these regulations, it's highly recommended that you follow them to control your risk, and I'll have more information on that later. There are also a few USP chapters that are specifically written for cell and gene therapy that can give useful information. So before I go to the challenges associated with cell and gene therapy manufacturing, I just want to stress the importance of applying or of having guidelines and good manufacturing practice for cell and gene therapy. The first one is to ensure patient safety. You really want to be able to control the risk of transmitting infection, impurities in the final product to make sure that patient is safe. You want to have an efficacious drug as well. You want to have product consistency. So you want to apply a series of rules so that you know your processes will give you a batch that has the intended quality attributes every time. And then obviously for regulatory compliance, because if you want to sell a drug, then you need to have the proper licenses. So, and the guidelines specific for cell and gene therapies were written because there are challenges that are more specific for that type of manufacturing. For example, starting materials are often human cells or tissues, which introduces the risk of variability in the quality of the material. And sometimes it's something you cannot directly control or that it's not fully understood, especially if it's patient material for autologous products. There's also a higher risk of having adventitious agents, so transmitting infectious disease through the starting material. So you need to have specific controls over it. The nature of the materials can also be a challenge for cell and gene therapy. They can be highly variable by nature, depending on their origin. Again, there's also a risk of having infectious agents in your materials. And the amount also can be a challenge for a testing program, for example. A lot of cell and gene therapy also involves the use of viral vectors to make genetic modifications because you don't control where they're going to be integrated. So you might have unwanted genetic modifications that can affect the efficacy of your product. And also sometimes the long-term safety is unknown because it's new therapies. The fact that a lot of these products are also intended to a specific recipient introduce a new risk. So if you have breaks, for example, in your chain identity, you can have product mix-up, and that can be very deleterious to the patient. So that should be controlled. And because these processes are fairly new, there's still a lot of open processing steps in the overall manufacturing processes. So that's an increased risk of contamination. These products can have a short shelf life that might not be compatible with the classic test methods, for example, USP 71, sorry. Sterility test that takes 14 days that might not be suitable for a product that needs to be delivered soon because it has a shelf life of 2 days. And also data integrity, so not all your equipment will be compliant with the requirements for data integrity. So that's another challenge. So let's go a bit more into details one by one. So probably the most important risk is the risk of contamination into your final product, because your starting material cannot be sterilized. Often your final product cannot be sterilized either. So it's really important that contamination control is control of contamination is very important. So Annex 1 and the Health Canada Guide 119 requires that you have a written document, a contamination control strategy that lists all the risk of contamination within your facility, and that you assess there's risk, that you make sure that you have the proper controls in place to mitigate that risk, and that you identify any potential gaps, and that you have a plan to control them. And you need to think about not only microbiological, so contamination of your product, but also cross-contamination. Some of the examples include the use of viral and non-viral work within the same facility. So regulators, by experience, the inspectors are really concerned about that type of activities within the same facility. So you need to make sure that your facility is designed properly to avoid the risk of cross-contamination. That there's no risk that somebody, that your vector will spread across your facility and contaminate a product that you don't want to be transformed by this vector. So think about the flow of material, the flow of personnel, how you control the fact that a person could potentially work on a viral vector and other products. Separate as much as possible the activities. Putting them on different HVAC systems is probably the safest way. Make sure you have good changeover procedure, making sure that you can eliminate any leftover products or viral vectors. Using dedicated equipment, especially equipment that cannot be decontaminated very efficiently, that have internal parts, should not be shared between viral and non-viral. And you might also want to consider having QC tests that can discriminate between two viral vectors if they're used in the same rooms or within the same vicinity. As I mentioned before, a lot of processing steps are still open in cell and gene therapy, which represents a risk of microbial contamination. So you should consider really having the right aseptic environment. Using isolators can also be a good idea here. Having good aseptic techniques, so your training program should show that your operators are well trained for aseptic techniques. Your aseptic process needs to be validated. Your environmental and personal monitoring program should be adequate also to detect any signs of contamination. And process design, try to close as much as possible your processes. Materials used in cell therapy also can represent a risk. So having a good materials management program in place, making sure that they don't represent risks that you're not ready to control. The use of single-use systems also is very important to prevent these risks. You need to validate, for example, if you have some filter sterilization during your process needs to be validated. And something you should consider also is the risk of introducing your your materials, because not everything is can be sterilized. So making sure that you have a good disinfection program when you transfer material into your aseptic areas. Again, your materials are also a source of risk that are a challenge for cell and gene therapy, starting with your starting materials that are a potential risk of propagating infectious disease, whether it's human cells or cell lines. As I mentioned earlier, following the cell tissue and organ transplantation guidelines is a very good thing when you deal with human tissues, especially for designing a screening and testing program to make sure that you reduce the risk of transmitting infectious disease. Cell lines should be also very well characterized in terms of potential virus, infectious markers that can be in there, but also if you have cells that have already been transfected with transfection material, you need to make sure that this is functional. And this is very well detailed in the ICHQ 5 chapters. The nature of the raw materials also represents a risk that's a bit unique to cell and gene therapy by their nature. Ways to control these risks is to use finished pharmaceutical products as raw materials. For example, if you need serum albumin in your process, sourcing them from a pharmaceutical company will reduce the risk because it's an approved drug. It's been tested, it's been validated already, and it decreases the risks. Trying to avoid materials that are from human or animal origins also reduces the risk. In the event that it's not possible, make sure that you qualify your supply and making sure that the risk of transmitting TSC and BSC is really well controlled. Using single-use format also is interesting because you want to avoid contaminating your product during the operations, and also you want to reduce the risk of cross-contamination. And establishing again testing programs if in the event that you cannot have such control raw materials, or if you have to rely on materials that are intended for research use only. Similarly, sourcing your disposables as a single-use system that are also medical device can reduce the risk. For example, if your syringe or needles is probably a good way to source medical furnitures as well. And again, your test plan should be designed in function of the risk that your materials pose to your system. So you should really well know the risk associated with your materials. Another challenge with cell and gene therapy is sometimes the release time is very short because the product is needed rapidly at the clinical site, whether it's because the product cannot be cryopreserved and has a short shelf life, or because of the nature of the disease it's intended for, that makes that the patient need it rapidly for treatment. So to control these risks, what you can do is design release process in a successive number of steps. So you can design that process so that you will have tests that are mandatory prior to shipment of the product, and then other tests that will come later before administration, for example. You should give priority on safety tests and ID testing. These should be done rapidly. And developing rapid microbiological detection tests is also valuable for such products. Keeping vein-to-vein traceability is very important, especially for products that are intended for a particular recipient. you need to make sure that you maintain the chain of identity throughout the whole cycle to make sure that there's no mix-up, that you provide the right product to the right patient. So you have to make sure that you can trace the cells or tissues from the donor to the recipient and vice versa. You also need to make sure that you control the risk of mix-ups in your facility from the moment that you receive the starting material to the moment you ship it out to the clinical sites. A good way to do that is to make sure that you have unique identifiers with at least two that follows the products throughout the process. Good labeling practices also, your product must be labeled at all time to make sure with these identifiers to reduce the risk of mix-up. And segregating your activities also is very important to make sure, again, that you are manipulating one product at a time to avoid mix-ups. The complex logistics also increases the risk. And these products are usually very sensitive to temperature excursions during storage, but also during transport. Again, because these products are precious and they are usually needed rapidly to the clinical site, you need to develop transportation routes that are efficient and validated, and maintaining a good chain of custody, making sure you can trace who had control over the product at all time is very important. And finally, data integrity can be an issue with electronic systems in the cell and gene therapy field, because not all equipment or instruments were designed to be compliant with the 21 CFR Part 11 or other regulations. Although I think it's less common now than it used to be at the beginning, there are still some instruments that are not completely compatible. So you need to make sure that you validate these systems. And if they're not totally compliant, you should have procedures in place to control the risks. Also protecting the raw data that is generated by these instruments by putting them in a secured server somehow sometimes can also be a challenge, because I found by experience inspectors and regulators pay more and more attention to this topic, especially for everything that comes from the lab. So now that we've seen the overall landscape of the regulations for cell and gene therapy, how do you prepare for an inspection? Something that you should begin to do way before your expansion is planned is know the risks that are associated with your processes and show that you have control over them. So risk and gap assessment here are very important to gain that knowledge. Also, it's possible that, by you find gaps during these analyses that you're not completely sure it's totally compliant with the regulations. In such cases, I strongly recommend that you prepare storyboards to present the inspectors. So instead of trying to convince an inspector that you're compliant, you can tell them that, yes, you're not totally compliant, explain the situation, but also say which controls you have already in place and what is your plan in the future to close these gaps. This will be especially once you come closer to your inspection date, this will be time that's better invested than trying to close these gaps, especially if you have bigger gaps. Being honest with the inspectors, but showing that you're still in control is very important. And I've put it there. I hated that phrase once I was first told it, but with time, I've realized that it's true, is that perception is reality. So you want to really show the inspectors that you're in control, that your processes are robust, and it starts by how your general facility looks. So make sure that your manufacturing areas, your labs, warehouse, offices, common space are clean and tidy. If you need like a fresh coat of paint can do miracles. If you have damaged walls, it will show that everything's well maintained. Do frequent tours of your facility, labs, with the managers, or send people from other departments doing these tours to identify anything that you think will attract the eye of the inspector. Make sure you don't have any uncontrolled documents laying around, that again, everything is clean and tidy. The day of the inspection, make sure you have somebody go around for a final tour. Empty the trash. Also, that's a very good advice somebody told me, because you never know what's going to be in their inspection, can find something that you don't want them to see in the trash. So make sure they're empty before the tour of the inspections. Identify and prepare your key SMEs. So you probably know which topics will be discussed during the inspections. So for these, identify SMEs within your team and prepare them and make sure they know the risks, they know your approach, and they can justify them so that they look confident in front of the inspectors. Having SMEs instead of having just the QA folks doing the inspection will show that you have the knowledge within the company, that it's not just quality that's responsible for compliance, but really it's a group effort that can reassure the inspections as well. And it's important also to train all the staff, because they can be questioned during the inspection by the inspectors. So train the staff from every department how to behave in front of inspectors. So make sure they know, they look confident, make sure they don't lie to the inspectors, that they rely on their procedures, and that they answer to the question that is asked. You want to avoid people that will go off track trying to answer a simple question and then give more information that they should have to an inspector. Another very important thing is to organize your inspection team. So identify the key people you want to have in the inspection room with the inspectors. Usually you want to have somebody that has a good overall knowledge of all the processes within the company. It's also good to have one writer per inspector that will write down the demands, but also that can write down the topics that are discussed with the inspector, so that that information can be transferred to the back room, so that people can start anticipating the demands and the topics that will be discussed later, and they can prepare accordingly in advance. Make sure you have an efficient system also of communication between the inspection room and the back room. And also your back room is very important, probably more important, I would say, than your inspection room. The stronger your back room is, the easiest it will be for you in the inspection room. So make sure you have people that can relay the information and the demands to all the departments. Make sure that team is able to review everything that will be brought into the inspection room before you actually see it. And you can designate one or two people that will be the only person that will come in the inspection room from the back room. So that being said, what does the current me think about all these guidelines? Well, if at first I found that it was very confusing, now I see them more like a roadmap. It's really a set of guidelines of instructions that you should use to orient yourself into that regulatory field, and make sure that all your processes are adequate, and that your risks that are inherent to your process are really controlled. And it's a very good way to make good products and make sure that the patients have the product they deserve.